Your DNA is a code. Look at all these nitrogen bases. 4 letters in what looks like a random sequence, but it's not. Your DNA code is a code to produce proteins. This video is on protein synthesis, and we're going to look at the 2 steps in protein synthesis: transcription and then translation. Transcription involves a DNA template strand. So this is just a piece of DNA on one of the 46 chromosomes. We call it a gene because it's a segment that codes for a specific protein. Now, a gene is actually much longer than this as far as the number of nitrogen bases, but this is just an example. So what we do is we take this template strand of T's, A's, C's, G's, and we produce an RNA molecule out of it based on the pairing rules. For example, what does T pair with? A. There's the A. What does A pair with? T. But remember, there are no T's in RNA. We would put a U there for uracil instead. C pairs with G, G pairs with C, and we can go all the way along this strand. And what we have done is we created an RNA molecule out of a DNA template. This DNA would have to unzip in order to expose the template strand. The RNA that we created is called a messenger RNA, because remember, the DNA is in the nucleus, but protein is made on ribosomes. They're in the cytoplasm, most on the endoplasmic reticulum, but there could be free ribosomes. So this messenger RNA that was created in the nucleus leaves the nucleus through the nuclear pores and enters the cytoplasm and finds a ribosome. Now the ribosome could take that messenger RNA and code a protein or create a protein out of it. That's called translation. So during translation, the ribosome will read the first 3 nitrogen bases, which is a code for an amino acid. Recall that amino acids are the building blocks of proteins. The next 3 nitrogen bases will represent another amino acid, and so on. So these pairs of 3 nitrogen bases each code for amino acid, which is the primary structure of a protein. Let's look at transcription in a little more detail. Here's one chromosome, but remember, when a cell is not dividing, it's in this long thread-like version that we call chromatin. And an enzyme called RNA polymerase will create the messenger RNA out of the DNA template. And here you can see how the DNA is unzipped to expose the template strand. So RNA is created by RNA polymerase based on our pairing rules. A goes with T, but in RNA it's U. T from DNA pairs with A, G pairs with C, and so on. So this is our messenger RNA that we created in the nucleus. This process is called transcription. Here we take a closer look at translation. Translation occurs at a ribosome. A segment of ribosome that's used to read the messenger RNA is called ribosomal RNA, or rRNA. It's written right here. It's the structural part of the ribosome. Here's the messenger RNA that's being fed through a ribosome. Remember that the ribosomes will read the messenger RNA 3 nitrogen bases at a time, and that codes for an amino acid. So when 3 nitrogen bases enter the ribosome, a transfer RNA, tRNA, will deliver the amino acid to the ribosome. That transfer RNA will leave, then the next 3 nitrogen bases will be read, and another transfer RNA will come in and deliver another amino acid. So you can see these amino acids being stacked on top of each other as this messenger RNA proceeds through the ribosome. So in translation, which occurs in the cytoplasm, transfer RNA delivers the amino acids to the rRNA, the ribosomal RNA, which is the ribosome, in order to grow the polypeptide, which is a series of amino acids which form the protein. So as a summary, in the nucleus, transcription occurs. That's where we create a messenger RNA out of the DNA template. RNA polymerase is the enzyme that produces the messenger RNA. In step 2, translation, the messenger RNA leaves the nucleus and finds a ribosome. Ribosomes are made up of ribosomal RNA, or rRNA. As the messenger RNA is fed through the ribosome, transfer RNA, or tRNA, reads 3 nitrogen bases at a time, which corresponds to an amino acid. And it will add 1 amino acid at a time as it's reading the 3 nitrogen bases being fed through. This grows the polypeptide chain, which will eventually produce a protein out of the ribosome. Step number 2 is called translation, and it involves all 3 RNAs. The messenger RNA from transcription, ribosomal RNA, which is part of the ribosome, and the transfer RNA, which delivers the amino acids to the ribosome.